So thank you very much. Thank you very much. And thank you very much to the local committee because it has been very interesting and the work that you have done, local committee. Thank you very much. So I start like this. And before I start with my presentation, I would like to all of you to stand up and to move slightly. Yes, please. It sounds strange. Sorry, I have a bias. So please stand up.
I know, we did it already one year.
Just stand a little bit, move your shoulders, just a little bit to behind, to the front, move your head slowly to one side and to another side, and just move the wrists. And maybe you think, why Esperanza is making us to do that?
But you are sure that you are not expert in public health?
Well, no, but I'm a yoga teacher also, so I cannot control myself when I see the faces a little. So
that counts in your time.
No problem, no problem. So move your shoulders behind, move your shoulders forward, and then the legs a little bit slowly. We are starting the day, and then just up and down, a little bit, up and down. Yes.
What's the less moderation for me?
Okay, very good. And then try to touch the ceiling a little bit. Yes, a little bit. Yes. Yes, and then we are almost done, Almost done. And if you can, try to touch your knees and then the floor, if you can. I'm not forcing nobody, so just, yeah. Okay, so now I think we can start. Please take your place. Okay, so thank you very much for doing this. Now I see you smiling, so I'm very glad of that. I'm also smiling, so thank you. So we are going through the microplastics. I will show you something different that we have seen during the day. We have had very interesting presentations during yesterday, so I will talk over ecotoxicology also. So the content of the presentation is microplastics in the environment, impact at different scales, the risk assessment, microplastic flow along the terrestrial food chain, microplastics assessment according to Minagri's project. So I will go there also, and some next steps. So during yesterday we have heard what is the pollution produced by plastic mulch, and also littering and the mismanagement of waste. So I'm not going further there. We had a very nice introductions. So also we know these slides, know the work of Sarker 2020 and also Sosa Machado. So we know that microplastics are in soil, are in the aggregates, and therefore the living beings that are in soil are also impacted. So, and I have this slide. I wanted also to show it briefly. So then, that we have primary sources, secondary sources of microplastics, and then how different organisms are impacted, invertebrates, invertebrates also, and different systems, agricultural systems. Where I put the stars is because in this presentation I will focus on those topics. Okay, I put this photo that was taken by one of my students in France. So why I put this photo? Because you can see what it seems like water is not water, it's plastic. And everything that is above is a kind of cucurbita. And then, and the forest nearby. So it's very paradoxical, no? Yesterday we heard, thanks to the presentation of Lea, that the fruits can be contaminated with plastics. So what we are seeing here, certainly those fruits this crop is also contaminated with plastic, and also below is contaminated. But then the thing is that the plastics are not static, they are also moving. So they are moving vertically and also horizontally. And then we have these forests nearby. So what is happening also with those places? Of course, we need more, we need to study more, but things like this we have in our planet in different places. So Okay, so how can we study the impact of microplastics? We can do it through different scales. So this is a diagram presented or done by Windsor et al. 2019. And then we have scales from ecosystem level and then community level, population, individual, and cellular and subcellular. So yesterday also we have seen examples of how microplastics can also affect on those levels. And then it occurs different processes and the effects. So the effects at the ecosystem level and also population, mortality, where we have seen that individual level, apoptosis, oxidative stress. So there are also posters that are showing how this occurs. So I think most of the investigations that we have nowadays are mainly from at the cellular level, individual level, more individual, population more or less, and I think we need to develop more studies related with community level and ecosystem level. So what is risk assessment? We have heard different definitions also yesterday, so I'm not going to go too deeply, but for doing risk assessment, so we need to have a problem identification, a target, a model. For instance, yesterday Luke explained us very well how the models can also give us insight of the problem of microplastics. So they can be used, and also we can do field and laboratory assessment, use of stressors, in this case microplastics, and then of course to do the risk assessment. Okay, so here I have another diagram. This is done by Bart Kuhlmans from Wageningen University. So I put it here because I find interesting how he's explaining also risk assessment. So to find the problem, and then to have a very good standardized method is important. Let me see how the pointing. Yes. Analytical method, standardized and to validate it, and then the effect. Yeah, effect, and then the exposure, and then to do the risk characterization. As I was mentioning, the plastics are not static. So these plastics are moving in agricultural systems and also in any system, in urban systems, in forests, in natural systems. The plastics are moving horizontally, as I was mentioning, and also go to the deep parts of the soil. And with the wind, they are coming with the water, by atmospheric deposition, I mean, and then also on the water, transported. And then invertebrates are also affected, and also vertebrates. Here are some cows. Yesterday also we had presentations over how livestock can be polluted with microplastics. So once that the microplastics are inside of the soil, yes, if the concentrations are not too high, then the invertebrates can transport it, ingest it and transport it, and when the concentrations are high, they die. Okay, so now I will present some examples in laboratory conditions. So according to Praveno 2022, these are like the variables that we need to, it's like the checking point, no? Like the variables that we need to pay into account, like for quality control. Most of the studies are focusing on the sampling, of course, what is the method, the strategy, and then polymers, particle shape, size, the particle number, of course. But here, this part, for instance, to have controls, we need to increase that more in our studies. According to this author, this is lacking a little bit, and also, clean air conditions. Most of the persons, when we do experiments, we try to have very clean conditions, but maybe to report more that. And then, Okay. So, G et al. 2021, after a metadata study, found that from this, the studies of ecotox with invertebrates has increased, of course, along the years. In 2020, there are more. And then most of the invertebrates that are studied are nematodes first, and then earthworms, and this is the type of polymers, and this is polystyrene mainly, but also we have studies with polyethylene, of course. And then here, the endpoint. Sorry that the diagram is smaller, but the endpoints, they did this study with 1,000, more than 1,000 endpoints and 56 studies. Yeah. Okay. Well, we know that microplastics produce an impact. So yeah, if the dose is high, then the invertebrates can die. But if it's not high, they transport the plastics in their feces, in their bodies, and then also they can deposit the plastics in the tunnels that they made, for instance, the earthworms or the other invertebrates in the biogenic structures. Okay. Different invertebrates, some photos here, also with fibers. It is a study done by Celonen 2020, and also a study done by Hodgson. Just examples that if you do studies with microplastics and invertebrates, yeah, there is an impact on them. Most of the time, of course, depends on the concentrations. If the concentration is very low, you don't see that clear effect also. That's important to mention. Okay, more related to the impact of the microplastics. So it is the damage of the tissue if there is ingestion, the ingestion range changes, and yeah, let me see here. The oxidative stress, yeah, there is an effect, oxidative stress, and also important to mention is, cytotoxicity, a study done with earthworms also by Kwak et al. 2021, that there are effects on reproduction and fragmented nanoplastic generation. So it's also important to say that most of the studies are not done with nanoplastics. We also need to increase more studies using nanoplastics, but it's understandable because it's difficult. But once that we have the good method, then to use them, of course. Okay, this is another example trying to see what is happening inside of the gut of the earthworms. This is a work done by Ke et al. 2023, and then he observes that inside of the gut and the different sections of the earthworm, there is the polymerization and there's physical fragmentation. Yes, he observed that in relation to the biomass, there is not a biomass change, but if there is a change in the ingestion. He did this study with oil-based plastic, but also bio-based plastics, and he sees a stronger effect with the bio-based plastics. So this is also important to try to understand that bio-based plastic, maybe we need to understand what is happening with this. And then he tested with different methods what is happening with the plastics inside of the earthworms, so by spectroscopy, but also by weight of the particles, the size of the particles, trying to understand in the whole system with the light density polyethylene, PBNT, and PLA. We know this slide, of course, done by De Sosa Machado 2019, so how is the effect of microplastics on plants? So there is a whole effect in the system inside, but yesterday we have heard also that in some experiments, depending on the plant, the type of plant, the type of the concentration of plastic, the type of plastic produce an effect. So those, we need to consider all these. So which type of species of plant, type of plastics. We need also to understand that we have a cocktail of plastics in the soil, but I will show you that after. And the type of soil. So there are different factors that we need to take into account when we are giving information, of course. But we are starting little by little. As at the beginning, in the presentation, well, not at the beginning, but during the presentation of Damnia, when he was saying that when he started to study microplastics, of course, was in another way, and after was more in detail, and the same with us, with the studies of ecotox. Yeah, first we wanted to see, okay, there is an effect of plastics. Well, we found that with high concentration, yes, we see an effect. Okay, so now we try to see with the smaller concentrations. Okay, So and first with the standard species, and then, but the thing is with different species there are different responses. Oh, pardon. Oh. Yes, okay. So then I will give an example for the selection of microbiome. I'm not going to talk of this because during the day today there are going to be presentations related of the effect of plastics in plants. Okay, so this is a study done by Ellie in 2021, and in this study they use soil from forests and with different types of oil-based plastics and different concentration also, and they saw that the microbiome is changing, the microorganisms, the diversity is changing with the different types of plastics, and also the abundance. This is very interesting to see that the PET is producing absence of abundance. So this is, yeah, and the PET, of course, we know these bottles that we have everywhere because we have since in the last 20 years everybody's drinking water from plastic bottles, so we have plastic bottles everywhere. So, and the interesting thing is this, yeah, well, and then in this experiment they observed a higher respiration with the high density polyethylene, if I'm not mistaken, no, with high density, and also production of cumulative CO2. But in the physical condition, it's more difficult to understand what is happening when it's plastics. So saltwater retention, water holding capacity, there are no significant differences among the treatments. Okay, so now I will give some examples for the field conditions. Okay, I think you have seen this slide, but as I was commenting to one colleague this morning, yes, studies of the impact on field conditions of microplastics on chicken, I have not seen another, so we observe this. If there is a mismanagement of wastes, then the microplastics can be ingested for invertebrates, and we find them in the casts of the earthworms, and then the chicken can be also polluted if there are microplastics. And of course, humans eat chicken. So, and we know already. We know already that if there is a problem with the mismanagement of wastes, then plastics are there. But also, if there are plastics mulch, we have heard studies already. So there is a transportation, there is an ingestion by the invertebrates, transported, and then affectation to the plants. But again, it depends on the concentration, the type of plastic, and the type of plant. And the chicken are accumulated. So what is a reality, independent of the type of plastic, is that the invertebrates and the vertebrates are polluted. And then it depends on the concentration, they can accumulate it more or less. But this is a fact. Okay, this is another example done by Nicolas Berriot. And then, yes, if the plastic mulch is not collected from the soil, then it can happen that, for instance, sheep, they can ingest the wastes of this plastic mulch. It's not very nice to see this, but it's happening. And then they can transport in their body these plastic particles to another places. So they counted 1,000 particles per kilogram of feces, so it is a lot. But fortunately, there are more studies now starting, for instance, in the University of Torino, that they are investigating the effect of plastics in livestock. In relation to the wildlife, also not too many studies are there. So we have this example that was done in Mexico, and then if there are landfills, then animals and the vultures in tropical places, these animals, they go to these landfills because we have created these landfills. So and then there are these garbage, and then they go there, and then they got polluted with plastics, of course. But the thing is that inside of them, in these pellets, there are also other substances. And then we know that the plastics can carry another substances, for instance, heavy metals, and also organic pollutants. So, and in this study, they saw this, that with the plastics, Borges et al. 2021, they found that there is this transportation. And these animals, they are not static, they fly, and they fly really big distances. This is just a close-up of this pellet, so you can see the different plastics that are there. So fibers are here, now inside of the pellets. Okay, so this is just a graph to say, okay, this is metadata done by Tunali 2023, and then this Tunali, he or she did this meta-analysis with the information collected, ecotox, risk assessment done around the world, and observed that the most is produced in Asia. Well, there are no significant difference with the works, the number of works that are done also in Europe. So these are like the areas of the world where most of the studies are done. And in relation to that, agricultural systems are the places that are like the places where are the factors where there's more pollution according to this metadata study. And also in North America, of course. But this is something, yesterday it was mentioned. It depends that in countries with more developed capacities, they can be more polluted, of course. But we don't have information from the other countries, so also we cannot do an affirmation on that. Yeah. So we need to do more studies. I think yesterday also was mentioned that microplastic tested in ecotoxicity essays, also by Tunali et al. 2023. So 77% is really, so 71% are more like fragments, yes, are fragments, and the type of plastics, 32% are PE, polyethylene. So we need to study also others, but mostly the cocktails. Okay, so here is, so this is a study that we did in 2023, and then we found in soil as field conditions that there are this cocktail of microplastics, that in soil there are 11 types of microplastics. So this is the example. Yeah, 11 types of microplastics where there are also biomulch, but of course in lower concentration. But this lower concentration is because they are just used or because they degrade. We need to understand what is happening here. And of course, PSC, mulch, we know that the farmers have used this PSC already 20 years, so there is a big concentration of this type of plastic. Of course, we have also polyethylene and the other types. Okay, in this study that we did of field conditions, we tried to understand if the management, for instance, the use of compost or plastic mulch produce an effect on the concentration of microplastics that are on different matrices, so for soil conditions, but also in sediment, in dish sediment, and also in airborne dust. And we found a strong correlation between the microplastics found in soils and microplastics in the dish sediment, but that was done by polypropylene. So we were thinking, okay, this polypropylene, where is coming from? We observed this mainly in those areas where they applied compost, but the highest concentration were found in those farms where they use plastic mulch. In relation to the compost, I will come back. In relation to the compost, not all composts are polluted. I need to be also clear with that, but we know that the compost, how cheap is the compost, how more polluted is with plastics. So, and the farmers know that already 20 years ago, 1980, they knew that already, at least in the Netherlands. Okay, so now in the same study, we observed also the soil invertebrates in these field conditions. And then, in the control systems, we found a higher diversity, more species of earthworms, compost lower, and then in those with plastic mulch, definitely not earthworms, but we found this larva coleoptera, and then we know that those are very resistant to different conditions. We know also with heavy metals that they can be very resistant to heavy metals, so it seems that also for plastics they can be. So we are trying to understand that, so to do experiments also in the lab. So, and here I put this graph just to show that Tunali, 2023, they did this risk assessment with metadata, and they say that Lumbricus terrestris, that is an endemic earthworm, is one of the species that is affected after this metadata that is like in risk. And it coincides with us, with our study, because we have these Lumbricus terrestris here in the control, but not when with the concentration of plastic is starting to be, then we don't have it. So it coincides. And why? Well, this is an anesic. That means that it moves from the bottom of the soil to the surface of the soil. So it is affected by the concentration of plastic that is inside the soil, but also what is on the surface. So it is very vulnerable. And the other species can tolerate more, a little, but yeah, at the end of the day, it depends on the concentration, again, and the type of plastic. Okay, so this is also meta analysis done by Wei et al. 2022. And then these are like endpoints that were more evaluated and where there are significant differences. So it's clear to see in some endpoints like basal respiration, movement index, reproduction rate, if possible to also it's possible to see an effect of the plastics on that. In relation to growth rate, it's more difficult, so that was not significant, and it's true, depends a lot on the concentration. When you have a very low concentration of plastics, you cannot see it, so then you need to assess it in another way. Mortality rate also depends on the concentration, type of plastic. And another factors, because also we do the studies and we use, this is stressor, plastics, but in nature we have also another stressors, for instance, climate change, there is a big inundation or it's too dry. Also those factors we need to put it into account, but it's more difficult to understand. And I say it also with experience. And I'm not presenting those results here, but yeah, it's also something to understand. Okay, so I come back to the work of Kuhlmann et al. 2020, and then we have seen some things. I invite you also to go to the posters. There are works related to oxidative stress and these aspects. Here, I'm not going to touch this aspect because it's going to be explained by Joanna today. Now I'm going to talk over Minagris, because also that was one of the objectives of this presentation, is what is Minagris doing in relation to ecotox. Okay, yeah. And then, so the aim of Minagris, well, you can see it is to understand the effect of microplastics related to agricultural systems. I invite you to go to the page to see more details, web packages, and also the deliverables that are public. Okay, so we have different plastics. These are photos given for different members of Minagris project, and then you can see plastic balls, so I'm not going to go there because, yeah, we know that. Okay, in the Minagris project, we have 11 case study sites, so those in red are the points, 11 case study sites in Europe, and then we are trying to assess microplastics at laboratory and field level. And we assess microplastics in soil. So what we did already, we had a field assessment campaign. So in these 11 case study sites, we took soil samples and we measured the microplastics, the number of types of microplastics. We are still trying to understand the behavior of this. So we are busy on that. But also we already started with experiments at lab conditions, and we are going to do also field experiment. So the field experiment, after this we have soil that we are using for our lab experiments are coming from the case study sites that have lower concentration of plastics. So and we are using this soil in all the places we are doing experiments. And for the field experiments, we are doing that in Wageningen, Dijon, and Craig. We are going to start next year with the experiments, but in Dijon, they already start something, so I will show. Okay, lab conditions for Minagris. So fast. Okay, so just to show you that we are working with the light density polyethylene, PBAT, starch, and also with another stressors, because as you know, in nature we have another stressors. So we are using antibiotic and also a pesticide or plant product, and then an antibiotic. And we are assessing different organisms and different concentrations. So earthworms, but also springtails. and also microorganisms, of course. Okay, so we are testing this, and just for example, different types of soils, different with several repetitions, of course, different species of earthworms, for instance, and also with the Polembola different species. Yeah. Okay, so just very preliminary results. This is, pardon, yes, and then this is in relation to the fungi. So it seems that there is an effect of polyethylene on fungi, and you can see these details on the poster that is done by Elisabeth Rasicot and Stefan Giese from Wageningen University. So I invite you to go there to see the poster. In relation to earthworms, I'm just showing in clay soil, it seems that a mixture of microplastic produces a bigger effect, a greater effect on earthworms, on Lumbricus rubellus, and the starch. So the starch is bio-based. So it seems that also is producing an effect. We need to understand more what is happening there. Okay. Also, just to show briefly, inside of the worms, also, the different plastics and with another stressors. We are trying to understand that also in the Minagris. So I'm also inviting you to go to the posters. There is a poster where it's showing that if you have different categories of earthworms, the behavior is different of the microbiome that is inside of them, endogates and anesics, and the microorganisms are really behaving different. So in the poster, and almost done. Okay, field conditions. So this is in Dijon, just to show you that we have different treatments with different concentrations, and we want to assess that in the 3 case study sites that I already mentioned. Okay, so next steps, fast, because Dimitrios is also saying Esperanza. Okay, so experimentation, of course, to continue to understand what is happening this at different levels, to communicate is very important. For instance, what I'm doing now, but also at different levels, you can also communicate also to your students. Workshops, we are going to have several workshops in Minagris, in each of the case study sites. Education is a key point. So we need to educate ourselves, but also educate the next generations and the older generations. We need to do a lot of education and engagement. And fast, I just want to mention the soil plastic app that you can download in your cell phone, and it's gratis, it's free. This was done by our partners in Austria. Here is Heide from Austria, and also from Spotteron. So you can assess macroplastics in soils. This is very nice for the students. They can also see, and help. This is all citizen science participation. And finally, this is what I have for you to present. So please go to the web page, register. You can get also the newsletter. There are interesting things there. So thank you very much for your attention.
Thank you for the nice presentation. Please.
Okay, Esperanza, thank you very much for the nice presentation. Unfortunately, we're running out of time, so if you have any questions for Esperanza, she will be around for the rest of the week, I think.
Yes.
But you have the time in the coffee break to ask questions. Thank you, Esperanza.
Okay, thank you.
Okay, so welcome. My name is